Autoimmunity and transplant immunology. Autoimmune diseases are humoral and cell-mediated immune responses directed towards self-antigens. They cause tissue and organ damage. They can be systemic, body-wide, or organ-specific. They're due to the loss of self-tolerance. In other words, the ability of the immune system to accept self-antigens and not initiate an immune response against them. So we have the development of T cells and B cells, and when those develop, all different types of receptors are going to be put on the surface, because essentially every T cell and every B cell needs to be able to recognize every single type of pathogen that we could possibly be exposed to in our life. So we have all of these different random combinations of receptors that are created, and sometimes those receptors that are created happen to recognize our own antigens as being foreign. But we have checks and balances. So in the thymus is where T cells develop, and in the bone marrow is where B cells develop. We ensure that they're not allowed to leave those areas if they want to attack us, our own tissues. We will let them leave if they tolerate us. In other words, if they leave our own tissues alone, they're allowed to leave those areas and progress to lymph nodes and other areas of the body or the lymphatic system, so they can wait and come in contact with the antigens that they're supposed to. But as we age, sometimes, for some reasons, and those reasons will be listed here in a little bit, we start to lose that self-tolerance, and we start to allow those T cells and B cells that recognize us as being foreign to slip through the cracks, and then they can start to cause some damage to our own tissues. So this is kind of a diagram. Again, I talked about the thymus, where T cells develop, the bone marrow, where B cells develop. In most cases, negative selection would mean that they recognize us as being foreign. That would lead to apoptosis. Sometimes those cells are allowed to differentiate into T regulatory cells, and then we have the T cells that can then be released. But if here we have a second check in the lymphatic system and they happen to recognize us as being foreign, then they would go through that process of apoptosis or programmed cell death. Over here, we have receptor editing, and then eventually they're allowed to leave and go to the lymphatic system. And if they do recognize us as being foreign, they would be killed. So there are genetic and environmental factors in the development of autoimmunity. Environmental and endogenous triggers, female hormones. Females are much more likely to develop autoimmune diseases than men. Tissue injury and release of self-antigens. Microbial infections, primarily molecular mimicry, epitope spreading, superantigens, the stimulation of immune responses. Epigenetic factors such as toxins, foods, drugs, aging, and post-translational modifications. There is also a genetically susceptible individual. They possess certain HLA or other genes. Autoimmune diseases typically run in families. So, for example, in my mom's side of the family, my mom has very severe autoimmune eczema. So it's not the allergic type necessarily, it's autoimmune eczema. And then my grandpa, her dad, had type 1 diabetes. One of my aunts has multiple sclerosis. Another one of my aunts has Graves' disease. I'm just kind of waiting to see which one I end up with someday. Hopefully none. But then you have the loss of immunologic tolerance, activation of self-reactive T cells and B cells, and development of immune responses to self-antigens. And that's what causes autoimmune diseases. With any term that you see, you try to break it down into its parts. So autoimmunity, auto means self. Immunity is your immune system is attacking yourself. So there's more than 100 autoimmune diseases discovered. Systemic diseases include systemic lupus erythematosus, rheumatoid arthritis, GPA, organ-specific autoimmune thyroid disease, type 1 diabetes, celiac disease, autoimmune liver diseases, multiple sclerosis, myasthenia gravis, and Goodpasture syndrome. So SLE, systemic lupus erythematosus, is a chronic systemic inflammatory disease that affects multiple organ systems. Patients develop numerous autoantibodies. For example, the antibodies against double-stranded DNA and other nuclear components, against lymphocytes, red blood cells, and platelets. Immune complexes form, so antibodies combine with those antigens and settle in the tissues. That triggers complement activation and the chemotaxis, or chemical movement in response to chemicals, of neutrophils and local inflammation. So antibody antigen complex settles in the tissues and it causes local inflammation. Clinical symptoms of SLE include joint involvement, skin rashes, renal involvement, neurological symptoms, anemia, leukopenia, thrombocytopenia, and others. Penia is a decrease in something. So leukopenia is a decrease in the number of white blood cells. Thrombocytopenia is a decrease in the number of thrombocytes, which is another word for platelets. Laboratory tests to diagnose SLE: a CBC, complete blood cell count, urinalysis, CRP, or C-reactive protein, ESR, erythrocyte sedimentation rate. Those are elevated during flare-ups. Complement quantitation, C3 levels, can be decreased during inflammation, and anti-nuclear antibodies. Anti-nuclear antibodies are directed against antigens in cell nuclei. They're present in more than 95% of lupus patients, include anti-double-stranded DNA, anti-single-stranded DNA, antihistones and nucleosomes, antibodies to the centromere or nucleolar components, and anti-ENA, such as anti-SM, RNP, SSA, SSB. The fluorescent ANA testing principle. So here we would have a patient sample evaluated, and we have labeled antibodies that are fluorescently labeled. So if the patient's sample has the ability to recognize certain types of antigens in the nucleus, the patient's antibody would then bind. And then we have this labeled antibody that we would add to it, and that would be able to detect whether the patient had those antibodies to begin with. And there's different results that we can see: rim, speckled, nucleolar, centromere, based on what type of ANA they have. Immunofluorescence using Cryptidia luciliae. This is an IIF using C. luciliae as a substrate. It contains circular organelle called a kinetoplast. It's rich in double-stranded DNA. It's used to detect antibodies to double-stranded DNA. So we put this down as an antigen, and then we would put the patient's serum on there to see if the patient has those antibodies against double-stranded DNA. And then we would put that secondary antibody on top of it, which is fluorescently labeled. And then we can determine if it's positive or not. There is an ENA Austerlani test. This is an immunodiffusion test that detects antibodies specific to ENA. Patient's serum or controls in the outer wells react with ENA antigens in the center well. In this example, patient A has antibody to SM. So here's anti-SM antibody, and here's anti-RNP antibody. Here's patient A, here's patient B, and here's ENA in the beginning. We look for lines of precipitation. So as this diffuses out, it's going to come in contact with ENA, and it has antibodies against ENA, so it's going to form this line of precipitation. Now it also has this line of precipitation with SM, but it does not form the complete line for RNP, so that one would be negative for RNP. Sample B is likely a negative control or negative for both because you're not seeing that line of precipitation forming. Phospholipid antibodies. Phospholipid antibodies are found in approximately 60% of lupus patients. They're associated with deep vein and arterial thrombosis, increased risk of recurrent pregnancy loss, can cause false positive results in non-treponemal tests for syphilis. Example, the lupus anticoagulant produces prolonged APTT and PT. Rheumatoid arthritis, or RA, chronic arthritis of the peripheral joints that can progress to joint deformity and disability. Before I go on, it's important to know the abbreviations for these. So SLE, systemic lupus erythematosus, also sometimes just called lupus, RA, rheumatoid arthritis. So going on here, about 25% of patients have osteoporosis. Some patients also develop subcutaneous nodules, pericarditis, interstitial lung disease, and vasculitis. Pathology and treatment of RA. Inflammation destroys the bone and cartilage. TNF-alpha plays a key role in the process. Overly active osteoclasts absorb the bone. So osteoblasts build bone, osteoclasts destroy the bone. They are stimulated by the parathyroid hormone to release calcium into the bloodstream. Autoantibodies combine with antigens to form immune complexes. Treatment would be NSAIDs, disease-modifying anti-rheumatic drugs, and biological agents that target TNF-alpha. Laboratory testing, we would look for rheumatoid factor. Rheumatoid factor is an autoantibody, usually IgM, that reacts with the Fc portion of IgG. It's found in approximately 80% of patients with RA, but it's not specific for RA. We can also look for anti-CCP, which is an autoantibody directed against cyclic citrullinated peptide, which is a peptide containing a modified arginine. This is highly specific for RA. We can also look for ANAs, ESR, CRP, and complement can be measured. Granulomatosis with pyoangiitis. This is a rare autoimmune disease involving inflammation of small to medium-sized blood vessels and respiratory tract. It progresses to more systematic disease involving other organs, such as the kidneys, joints, and skin. Most patients have antibodies to neutrophil cytoplasmic antigens, such as proteinase 3. Neutrophil activation results in damage to vascular endothelium and a Th1 response. Anti-neutrophil cytoplasmic antibodies are produced against proteins in neutrophil granules, strongly associated with syndromes involving vascular inflammation, detected by IAF on ethanol-fixed leukocytes, ELISA or CLIA. And this is an image of the fluorescence. Autoimmune thyroid diseases. Thyroid hormone synthesis is carefully regulated by endocrine feedback loop. Autoantibodies produced in AITDS can lead to a decrease or increase in the production of thyroid hormones. Hashimoto's thyroiditis is immune destruction of the thyroid gland and produces hypothyroidism. Hypo means less or decreased function of the thyroid. Symptoms include fatigue, dry skin, weight gain, brittle hair, and the formation of a goiter. Laboratory results, normal or high TSH. TSH is thyroid stimulating hormone. So either there's no effect on TSH or TSH would be high because it's trying to stimulate this thyroid that's basically dying. Low free T4, anti-TPO, or anti-TG. Graves' disease is the opposite. So this is an AITDS characterized by hyperthyroidism. Symptoms include nervousness, weight loss, rapid heartbeat, goiter, and exophthalmos, or bulging of the eyes. TR antibodies are produced. You have low TSH and high FT4. Antibodies to TPO and TG. Type 1 diabetes mellitus. It's an endocrine disorder characterized by hyperglycemia, so a lot of sugar in the blood. Type 1 destruction of beta cells in the pancreas results in insulin deficiency. Long-term effects: you can have cardiovascular disease, kidney dysfunction, nerve damage, blindness, and infections. Laboratory results would be increased glucose blood level, elevated HbA1c, that's glycated hemoglobin, so glucose particles on the hemoglobin molecules, autoantibodies to GAD and IA2 and ICA. Celiac disease affects the small intestine and other organs. It's triggered by gluten. There's autoantibodies form in HLA-DQ2 or HLA-DQ8. Positive people to gliadin, tissue transglutamase, or endomysium. Symptoms include diarrhea, abdominal pain, and bloating. Treatment would just be to follow a gluten-free diet. Autoimmune liver diseases, immune-mediated liver disease that can lead to end-stage liver failure if left untreated. Clinical features include elevated liver enzymes, fatigue, nausea and vomiting, abdominal pain, itchy skin, jaundice. Autoimmune hepatitis, or AIH, the hepatocytes are targeted. AIH1 positive for SMA or ANAs, and AIH2 is positive for LKM1 or LC1 antibodies. Primary biliary cirrhosis, or PBC, is the destruction of intrahepatic bile ducts and cholestasis. Majority of patients produce mitochondrial antibodies, or AMAs. Multiple sclerosis. So this is an autoimmune disorder that involves inflammation and destruction of the central nervous system, particularly the oligodendrocytes in the brain and around the spinal cord. Most patients produce antibodies against myelin basic protein, which makes up those cells. Plaques form in the white matter of the brain and spinal cord, causing the destruction of myelin sheath of axons. Symptoms include visual disturbances, weakness in the limbs, dizziness, and sensory abnormalities. These symptoms can come and go. They can be persistent, they can be chronic, and they can be progressive. It all depends on how severe it is in that individual person. Laboratory findings: lesions seen on an MRI, or magnetic resonance imaging, increased immunoglobulins in spinal fluid and increased IgG index, oligoclonal bands on protein electrophoresis of CSF. The unfortunate thing is there isn't a simple blood test that can say, yep, it's multiple sclerosis. So it usually takes many years and a lot of consistent symptoms and visits to the doctor before a definitive diagnosis can be made. Myasthenia gravis affects the neuromuscular junction, resulting in weak skeletal muscles. Most patients have antibodies to AChR, which blocks the binding of ACH, or acetylcholine, to its receptor, and transmission of nerve impulses that activates muscles. So you have decreased transmission of those signals and decreased activation of muscles. Goodpasture syndrome. Patients produce autoantibodies to basement membranes lining the renal glomeruli and lung alveoli, specifically directed against alpha-3 chains of type 4 collagen. Immune complexes bind to basement membranes, attract complement, and cause damage by type 2 hypersensitivity. The syndrome can progress to renal failure and respiratory problems, cough, shortness of breath, hemoptysis. Antibodies to GBM found in most patients, detected by IIF on frozen kidney sections or ELISA for antibody to alpha-3 subunit. Tissue-bound anti-GBM detected by direct immunofluorescence on kidney biopsy sections produces a small linear or smooth linear ribbon-like fluorescence along GBM. you can review the summary slides for the autoimmunity, and we're going to continue on to talk about histocompatibility and transplantation immunology. So transplanted organs have foreign antigen. Large enough organs carry with them foreign white blood cells or foreign inflammatory mediators. So there's different things that can happen when we do transplantations. But transplantations and whether they're going to be compatible or not has to do with histocompatibility systems. One of the major ones is the major histocompatibility complex, MHC. We talked about this previously in this course, but this is closely linked to genes on chromosome 6. It's highly polymorphic and induces strong graft rejection. There's class 1 proteins that are expressed on all nucleated cells and class 2 proteins that are expressed on antigen-presenting cells. Other systems involved in graft rejection would be the minor histocompatibility antigens, MICA antigens, ABO blood group antigens, and KIRs. The inheritance of HLA haplotypes differs, and you can kind of see the rearrangements and how they can be inherited. And the types of grafts also differs. So we have autograft, isograft, allograft, xenograft. An autograft is tissue transfer on the same individual. And every time I talk about autografts, I always have to bring up my uncle. So my uncle had very significant dental problems, and he had this painful tooth that toothache would not go away. So he went to the dentist, and they discovered that it wasn't just a toothache. It was bone cancer, and the bone cancer had spread deep inside of his jaw. And that bone cancer actually was had metastasized from an original lung cancer. So he was a smoker, and he smoked for a great deal of his life and ended up getting lung cancer, and that lung cancer metastasized to the oral cavity, and he got bone cancer in that the mandible, and he had to have all of that removed. So he had a big chunk of his jaw removed, a portion of his lung. He had to have lymph nodes removed from several areas, including areas around the lungs and in the neck, all the way leading up to where the cancer was in his jaw. So they had to do some rerouting of blood vessels around in his body. They had to take a blood vessel from his leg and route it up to restore the blood flow in his face so that his face muscle tissue and everything could still be alive. And it was a major surgery. And it was a pretty intense moment. He has been cancer-free for like almost 25 years now, so the operation worked and all the radiation that followed worked, but he was really sick for a long time. But that's autografting, taking tissues and components from one part of the body and moving it around on the same part of the body, or a different part of the body on the same person. Isografts has to do with grafts or transplantations between genetically identical individuals. So this could only happen between identical twins. An allograft would be a transplant between members of the same species. So if I were to donate a kidney to my mother, then that would be an allograft. So we are not genetically identical. We are from the same species, though. Xenograft would be transplant into members of different species. So if we were to transplant a pig valve into a human heart, for example. There's direct versus indirect allorecognition. So direct would be cytotoxic T cells from the recipient bind directly to foreign HLA antigens on the cells of the allograft, and they release cytotoxic factors. So cytotoxic T cells from the person who's getting the organ bind to the incoming foreign antigen. Indirect would be host antigen presenting cells present foreign MHC antigens on graft cells to recipient helper T cells. So here is direct. Host cytotoxic T cell binds to and recognizes that graft cell, leads to apoptosis. Indirect, the host antigen presenting cell processes the graft's MHC presents that to a helper T cell, and that ends up with releasing cytokines, and cell-mediated cytotoxicity, antibody-mediated reactions occur. There's different types of graft rejection. So hyperacute occurs minutes to hours after the transplant. Preformed or preformed antibodies to blood typing, like ABO antigens, HLA and endothelial antigens bind to vascular endothelium and activate complement and clotting factors. Accelerated occurs several days after the transplant. Mechanisms same as hyperacute. Acute occur months to days or days to months after the transplant. Cytokine production by the helper T cell induces delayed type hypersensitivity. CD8 T cells mediate cytotoxicity, and antibodies bind to complement and induce inflammation. And then chronic is when you have rejection 1 year or more after the transplant, and that is a delayed type hypersensitivity reaction. There's also graft versus host disease. In graft versus host disease, lymphoid cells in a graft start mounting an immune response against the recipient's histocompatibility antigens. It's caused by immune response of T cells in the donor HSC, lung, or liver transplants. Involves massive cytokine release, inflammation, and destruction of various tissues. Occurs 100 days or more after the transplant. Immunosuppressive drugs are listed here. Corticosteroids are primarily used, antimetabolites, calcineurin inhibitors, monoclonal antibodies, polyclonal antibodies. And HLA typing can prevent graft rejection. HLA typing is the identification of HLA antigens or genes in transplant candidate or donor. Complement-dependent cytotoxicity tests, HLA antigens identified by their reactivity with panels of antisera. There's also HLA genotyping, which are molecular methods used to identify the HLA alleles. The principle of the CDC test, so here you can see the reaction happening. HLA genotyping, you would get the identification of those alleles. It has advantages over CDC, higher resolution, the reagent quality, and high throughput. And then there's molecular methods, the PCR. So in PCR, we have a very specific target sequence. We have a forward primer, a reverse primer, and if the sequence that we're looking for is present, it would make a copy of that. And when we run that out on gel electrophoresis, we would be able to see that copy out on the gel. If it didn't have that gene, there wouldn't be any application of that gene, and we would run it out on the gel and see nothing. Then there's also probes that can be used. So we put a probe in a well, and if the amplicon is amplified, it would be linked to a signal, and you'd see a color change in that well. HLA antibody screening and identification. So HLA antibodies may develop in response to multiple blood transfusions, prior organ transplants, paternally derived fetal antigens by pregnant women. These antibodies can cause hyperacute graft rejection. Donors with the HLA antigens that can react with the antibodies in a transplant candidate should be eliminated from consideration. Testing HLA typing by the CDC method. Patient serum is incubated with panels of lymphocytes. Anti-human IgG can increase test sensitivity. Percent panel reactive antibody is reported. ELISA uses purified HLA genes bound to microtiter plate wells. Flow cytometry or cross-matching. After a suitable donor is identified for transplant candidate, a donor-recipient CDC test is done to confirm absence of donor-specific antibody. There's also flow cytometry for HLA antibodies. Patient serum is incubated with a mixture of beads coated with different HLA proteins. You wash and add an FITC labeled anti-IgG. You wash and analyze for fluorescence on a flow cytometer. And you can read through the summary.